Good afternoon to all who have just joined us at TechInnovation 2020. The next program will be a range of crowd pitching presentations on the theme Health and Personal Care. We have lined up 7 speakers from leading corporates who will pitch their medtech and biotech technologies to enterprise partners for commercialization opportunities in health and personal care. Each speaker will be given 5 minutes to present. There will be no Q&A following each presentation. Should you have any questions during the presentation, please type them on the Pigeonhole Q&A platform by scanning the QR code at the top right corner of your screen or visiting the website at pigeonhole.at/techinno2. Alternatively, you may wish to visit their digital booth at techinnovation.com.sg. Without further ado, I'd like to introduce the first speaker, Dr. Tun Yun Lee, professor at the Hanyang University.
Hi, everyone. Nice to meet you. My name is Dong Yun Lee. First of all, I would like to appreciate the community of tech innovation for giving me this opportunity. Today, I want to introduce a technology that can detect tear glucose on eye. So as you know, diabetes mellitus is one of the world's top 3 diseases and causes many complications. Therefore, in order to manage diabetes disease without complications, it's necessary to continuously monitor blood glucose level. Currently, blood glucose monitoring is performed using the finger prick method and the implantable glucometer method. In order to minimize the patient convenience or the various discomforts, this invasive method is being extended to non-invasive method in the future. Representatively, the smart contact lens and mouthpiece sensors or the patch type sensors are being developed. In my case, I researching a contact lens type sensors. When developing contact lens type sensor, the first consideration thing is the concentration of glucose in tears. As shown in the table, the level of glucose in tear is about 3 to 10 milligrams per deciliter, which is more than 10 times lower than blood glucose level of 80 to 90 milligrams per deciliter. Also, the difference in the level of glucose tears of the normal and diabetes patient is not significant. Therefore, the contact lens type sensor must have the responsiveness and accuracy to detect low concentration of glucose in tears. So, in addition, in order to detect low concentration of glucose in tear with high sensitivity and reliability, it's being developed with the electrochemical contact lens type sensor. These electrochemical contact lens type sensors are introduced with IC chips and wireless antennas, and the battery package must also be included to drive them. As a result, the electrochemical contact lens sensors can generate heat while driving, and there is a risk of damage to corneal tissue of the eye. In addition, a complex manufacturing the process makes a contact lens type sensor expensive. So the method developed by our team is developed a nanochromophore that can develop color by reaching with glucose in tears. And its manufacturing process itself can be manufactured by mixing nanochromophores with material during the conventional manufacturing process. In addition, to quantitatively analyze the color represented by the nanochromophore in the contact lens, and algorithms, including normalization of variables, was developed, which was designed to be analyzed using camera module of the smartphone. As a result, as you can see, in the movie, the sensitivity to glucose was able to differentiate between 0.1 millimole to 0.6 millimole. So captured images that are selected and analyzed with these algorithms. So in fact, this colorimetrics contact level types sensor was won the eye of the rabbit with diabetes mellitus to check the possibility of color development. As a result, it was won the eye of the rabbit for 30-minute analyzed. So we can analyze diabetes levels of the rabbit. In addition, the human volunteer tear samples were exactly monitored. So these levels with same patterns when compared with conventional glucose assay kit and the portable glucose meter. So I'm looking for these collaborators for these redesign of contact lens and CRO validation, GMP grade production, clinical trials. Okay, thank you for your attention.
Thank you very much, Dr. Tung Yun Lee. Next, from Mind and Tech Limited, we have Fan Si Wing.
Hi, I'm Wing from Mind. In these 5 minutes, I would like to introduce our company to you and the Alzheimer's disease detection kit with the Nano innovation. At the end of my presentation, I hope you can know more about our company technology and give us advice in the Q&A section. Alzheimer's disease is the most common form of neurodegenerative dementia that will progressively destroy memory, thinking skill, and eventually low down our sense of behavior. There will be 1 in 3 people over the age of 85 have the Alzheimer's. In 2015, there is around 13 million suffer from the disease, and the number is projected to be triple in 2030. The cost of the dementia care is increasing with time worldwide. However, there is no single confirmative test to diagnose AD until now. The cause and the progression of the disease is unknown, and it is incurable. The current Alzheimer's disease diagnostic method, including the number puncture, which will collect the CFS from your spinal cord, or the PET scan, is neither invasive or expensive. Our company, Mind and Tech, led by Professor Wang Shen and Professor Li Hongwing, would aiming to provide a cost-effective, simple, direct, yet sensitive platform for early neurodegenerative disease detection and diagnostic. Our R&D team has been working in the area of disease diagnostic for over 10 years. We have published a research article in the scientific platform and journals in newspaper. Our innovation has been patent protected. Our company would like to offer detection kit, solution for AD diagnostic, and the MRI contrast agent. Recently, we are focusing on developing the testing kit. The principle of our detection kit is to detect the concentration of circulating biomarkers in the serum, urine, and the saliva. This is our first generation laboratory use detection kit for beta amyloid detection, and it is now available in our website. There is several benefits of our kit. First, we use simple and conventional instrument to test and can provide the result after 1.5 hours. Second, it is cost effective that we only require $100 only. It is 15 times lower than the current diagnostic method. Third, it is non-invasive that we only need 1 drop of your blood or your saliva or your urine. And the sensitivity of our detection kit is high. This is the scheme of our innovative kit. And we have worked on the clinical serum sample. From the data, it shows that our detection kit can capable to differentiate the AD patient from the healthy control. In the coming future, we would like to invite R&D collaborators to help us develop the second generation of detection kit for the tau and the p-tau, and finally generate a point of care tool for the with validation. And we would like to also gather investors helping us to line up with hospital to carry out the clinical trial and verify with standard method, and also provide us the clinical sample, and in order to explore the application for the EU and FDA approval. For our new launched product, we would like to optimize our product by the packaging redesign and the marketing. We are willing to connect our customer and user through the similar as the elderly center or to the medical professionals. We will also like to attend the conference as well as the entrepreneurship workshop in order to promote our diagnostic kit and introduce more about the AD to the public. If you are interested in our innovation, hope to see you at the Tech Innovation meeting. Thank you for your time.
Thank you very much, Wing. Our next presenter is from RVmagnetics. This is Henrich Tauber.
Dear ladies and gentlemen, my name is Henrich Tauber, and today I would like to present you the smallest passive sensor in the world. So just to give you an overview of our company, we are an R&D company based in Slovakia, European Union. We've been established in 2015 with a strong scientific and business background, where the R&D team consists of all engineers, and they have earned their PhDs from the microvirus, which is our sensor that I would like to discuss today. So please let me allow you to introduce you, based on more than 25 years of research in applied magnetism, the smallest passive sensor, the microvirus sensor. As you can see on the screens, it is very thin size, even thinner than a human hair size sensor that can be embedded into objects. It provides direct, contactless measurements on pressure, temperature, and magnetic field, indirect measurement on stress, movement, vibration, heartbeats, 3D positioning, flow, electric currents. As you can see, it's very small, even 1 centimeter in length and 20 microns in diameter. Looking at the features and the benefits of overall, it provides you with the micro dimensions that can be embedded into different applications, from material to human body. It provides contactless measurements as it is a magnetic sensor. It is multifunctional, durable, and versatile. It provides real-time sensing, a long lifespan, and passivity. With the combination of all of these, we can customize it specifically for you to provide a tailor-made solution. In healthcare and medical technologies, this sensor is thinner than nerves. It is biocompatible due to the Pyrex class that we are using. It has no effect on X-ray, and there's very minor effect on MRI. And it is even though it is a metallic sensor, it still has minor effect on MRI, as you can see on the screen. Looking at the specific use cases, one of them is a smart petri dish, and we have developed a Bluetooth in vitro fertilization contactless device to ensure exact temperature, because it is so crucial for success for IVF. We are actually helping to control and maintain the temperature with a sensitivity down to 0.01. And all we are doing this with non-invasive contactless measurements. Right now, we are developing this application with an undisclosed medical partner. Also, from the temperature, we are providing identification of a specific petri dish, something like an ID tag. Moving on to other projects within the healthcare industry, we conducted different applications, from the drug delivery devices to acne monitoring of titanium implants, also localization of abnormalities in human body, smart mattress, or smart wearables. This sensor is versatile. It can be used in many areas, not only in healthcare. As you can see, the business focus from healthcare is in 5 other verticals. Business verticals. So one of them is electric motors, embedding it, getting the temperature from inside the motor, transportation logistics, composite materials, so predictive maintenance and structural health monitoring of different materials, thermal management of batteries, and last but not least, construction and civil engineering. If you look at our business needs, we're looking for R&D collaboration for an adopter, client, or integrator to productize fully developed technology. So my question to you is, what are your needs? We are not offering off-the-shelf solution. The sensor is for customizable R&D collaboration. So we need to understand, what are your needs? Is it a sparkless sensor, a biocompatible sensor? Is it a sensor for smart wearables? Or do you have some limitation regarding the weight? Or the environment is very electric, there are very strong electric and magnetic fields. So once we understand a little bit more about your needs, we can customize it and fully develop a prototype and product for you. So should you have any questions or would like to understand how it actually works, this magnetic sensor, I'll be more than happy to answer your questions through my email or just contact me directly through the tech offer. Thank you, TechInnovation, for this opportunity, and good luck to all of you.
Thank you very much, Henrich. We would like to remind everyone to type any questions you may have about the presentation thus far on our Pigeonhole Q&A platform by scanning the QR code at the top right-hand corner of your screen or visiting the website at pigeonhole.at/techinno2. Next, from Preppers, we have Dr. Shuhei Aramaki.
Hi everyone, I'm Shuhei Aramaki, a doctor in charge of sales development at Preppers Incorporated. The presentation titled High Resolution Mass Spectrometry Imaging and Selective Reaction Monitoring Imaging. First, let me introduce the general workflow of MSI. We make a cross-section sample from an animal or human and place it on the glass slide. After these preparations, the sample is measured with MSI. If using MALDI, the glass slides must be ITU coated and the matrix must be applied before the measurement. Using DEGI and MALDI tools, we can detect thousands of analytes from a sample and obtain their distribution faster than any other method. Furthermore, this data can be measured without invasive manipulation or radioisotope labeling, another feature of MSI. Next, let's talk about high mass resolution MSI. High mass resolution MSI, such as Solix, XR, can clearly detect stable isotopes and even molecular mass differences in a few milldalton. The difference in molecular mass can be expressed as imaging. For example, in Figure B, the difference in mass is detected by 3 milldalton, and the difference is shown as imaging in Figure A. Finally, I want to talk about selective reaction monitoring, MSI. SRM allows us to monitor a specific molecule. As shown in the figure, the first thing it does is single out the large M2 from among the rest of the ions. However, this is not enough to distinguish between the same mass substances, so we use collision-induced dissociation, CID, to break down the large M2 into smaller pieces by colliding it with the gas, and then selectively detect only small M2 in the fragments that appear. This makes it possible to selectively monitor only fragments of small M2 materials with large M2 mass. For example, this is an experiment to determine the distribution of lecithin, a component of Chinese medicine, in the body. Since lecithin is known to change its M-obesity transition from 417 to 255, we only detect this change by SRM. Preppers Incorporated can offer mass spectrometry technologies, and we are composed of medical doctors and mass spectrometry specialists so that we can handle human specimens. Utilizing these human resources, we are currently developing medical devices. Using mass spectrometry technology, please contact us if you are interested in the medical development applications of mass spectrometry. Thank you very much for your attention.
Thank you very much, Dr. Shuhei Aramaki. Next, from the City University of Hong Kong, we have Dr. Shen Yajing.
So, good afternoon, everyone. This is Yajing from City University of Hong Kong, and I would like to share my recent research of the development of Millirobot for medical applications. So the idea is, in the biomedical engineering field, we have a lot of things to handle. For example, we need to move unconstructed object into our body, like the pills, like the capsule. And also we need to move something just like the catheter inside our body. And on the other hand, in our body is very complex. And the environment are totally different in the stomach, in the blood vessel, or in the gut. So in this case, one key problem here is how to manipulate such kind of small things effectively. Okay, so we proposed one solution. Our solution is to develop one kind of glue, and such kind of glue can convert the small object to the robot directly, as shown in this image, just by spotting a few of glue on the surface of the teeny object. Then, under the application of a magnetic field, we can control its motion remotely. Here I have some video to show you, to share how it works. So just by spraying a little glue on it, and after curving, we can use the magnetic field to control its motion. And as to the biomedical application, we think first we can change the catheter from a negative catheter to a positive, to an active one. And then it can be used to some surgery. And also, we think we can apply this kind of technique to develop some active drug delivery system. As shown here, for example, we can cover the capsule, and we then take the capsule to the targeted position, and then to release the drugs. Okay, and for the corporation, we first of all, we hope we can have some research and development cooperation, because this research is at the preliminary stage. We just published a paper in the top journal, and we need more to work on this. For example, how to handle a specific disease, right? How to cooperate with the doctor and some other things. And also we hope we also can transfer the pattern about this technique. We already applied the pattern, and if you are interested in this one, we also can do some cooperation in this. And lastly, if you have chance to make some starting up company and investment, you are also welcome. So that's all for my presentation. If you are interested, we have a discussion later, and also can send an email to me by this address. Thank you.
Thank you very much, Dr. Shen. Next, we have Chong Hui from JAH Life Pte Ltd.
Hi, everyone. Thank you for your time. I'd like to quickly introduce ourselves. Today, my name is Chong Hui. I'm going to introduce my company, JAH Life, and also the product that we have developed. It's called SanitatemAlpha, which is a multifunctional cosmetic active. Okay, too many words, so I'm just going to sum it up. JAH Life is actually a spin-off that we started this year in midst of COVID in April. That's exactly 1 year anniversary since the founding of JAH Tech, which is the holding company. This is one of the 3 companies we have spun off. And this company, JAH Life, is focused on technology related to skincare and skin-related healthcare beauty products. Okay, so what is the problem that we saw in the market? Process that we are able to develop and bring about things like carbon, water, and sodium, which is sodium chloride, together into a single emulsion. And what we found is some of the key properties of Sanitatem is as follows. So it has displayed anti-aging properties. It's definitely not irritant. Its anti-inflammation properties also helps calm down the skin. So things like brightening and antioxidant. So by combining all these multiple actives together into one single element, it makes it a very ideal ingredient to use for a lot of formulators. So some advantage of our technologies, as you can see here, is that we have spent many years in the labs to develop this, and the efficacy are comparable or slightly below or near to those actives that are available out in the market. And it's very easy formulated as a base into products, because the majority of the product is made out of aqua, water. So it's a very simple to use base. Of course, we have tested it with third party. I have our dear partners and friends introduced by our friends from IPI, Denova Science. They have tested this product independently and has given us all the test results. So if anyone's interested in those results, we can actually share with you. Okay, so our technology is ready, and we are already being incorporated into multiple products, both in Singapore, Hong Kong, China, Korea. So what we are looking for is, of course, I have the products ready, as in the Sanitatem, right in my office right now. So if you are looking to maybe test it out, just give me a call and I can pass out the samples to you. And if this is something that you you'd like to use, you can also definitely form an arrangement where you can place in large orders. So far this year, we have already shipped over 2,000 liters to users and manufacturers in Korea. Technology licensing, that's another potential collaboration method. And finally, will be partnership and joint venture. Thank you for your time. If there's any question, please feel free to contact me here.
Thank you very much, Chong Hui. Our final presenter for this segment is Masaharu Takahashi from Goryo Chemical Incorporated.
All right, would like to introduce our in vivo navigation drug, that is active visualization technology for intracorporeal surgical operation. Navigation drug, shortly say ND, is our platform technology visualizing specific target materials, including cancer cells, secretions, and molecules intracorporally as well as extracorporally, on the basis of site-specific active and strong luminescence fluorescence probes. The LD is designed to start emitting fluorescence through chemical reactions on receptive substance of the target materials, but shows almost no fluorescence before the reactions. The activated LD's strong luminescence provides clear fluorescence recognitions due to the usage of specific fluorophore and chemical linkage technology. In case of cancer surgery, the ND not only decreases cancer cell remaining and improves patient's quality of life due to accelerating discriminant between cancer and normal cells through surgery, but also makes the total surgical time shortened and improves operational productivity due to providing real-time pathological diagnosis to operation teams. In case of digestive organ surgery, the ND could visualize auxiliary risk factors such as pancreatic fistula and improve the operational productivity. The ND technology has great potential to cover any indication of diagnosis and surgical treatment on the basis of detecting the specific target biophysical materials. Regarding ex vivo ND, we already completed one clinical trial of GCP-001 for breast cancer detection and are just about to achieving marketing approval. Since we are going to have a sales alliance regarding the ex vivo LD in Japan, we seek out licensing partners on the other regions. Regarding in vivo LDs, we already started one clinical trial of GCP002 for esophageal cancer detection and some preclinical trial, including GCP003 for pancreatic fistula detection. The GCP002 is under investigation of clinical safety and effectiveness trial in operational environment for the detection of esophageal cancer under endoscopic surgery and is in the process of trying to achieve the clinical POC. The strong fluorescence became active within several minutes after spreading the LD on the target materials. The surgical operation team members can perform the operation safely and accurately with real-time pathological diagnosis, and the visibility of the residual cancer contributes to reduce the risk that the cancer region will remain unresected. We seek out-licensing or co-developing partners of the GCP002 as well as fluorescence detection devices for use with the GCP002. Further, we also seek strategic partners to collaborate on the platform-based development approach for new in vivo diagnostics, including GCP003. Thank you.
Thank you very much, Masaharu-san. We've come to the end of the first crowd pitching session of the day on health and personal care. Thank you to our speakers and to all attending this webinar for your active participation today.